the three of us are people that are mostly doing theoretical work, not hands-on parts. That part is handled and provided by Professor Arda Denzi Alçınkaya from the EE department, Sema Dumanlı Oktar, who's also from the EE department in our university, and Cansu Canbek Özdil, who is just starting as a very young professor at Yeditepe University. Among the others, the first three were myself and Birkan are from the computer engineering department, and Ali Emre Pusani is also from the EE department at Boğaziçi University. Sorry. So very briefly, what is nano networking, and from there we will come to molecular communications. First of all, about the nano machines. Typically, a nano machine is a device which has some components at the nanoscale, and of course, it's supposed to be doing some useful function as a machine. That machine could be a human-made machine or an engineered cell, or maybe a combination of these. From our perspective, the important things are at least one of the dimensions should be in the nanoscale. That's a requirement of being called a nano-networking device by the IEEE P1906.1 standard group. It should be human-controlled, otherwise all living cells are actually satisfying the first rule. It should be able to process data and be capable of communicating by utilizing some carriers of information at the nanoscale. The standard group, of which I'm a member, defines the nanoscale as 100 nanometers or below. At least one of the dimensions should be at 100 nanometers or below. Other dimensions could be larger. So this actually, we gave this flexibility to allow for devices that could work also as gateways between the nanoscale and higher scales. So nano machines, of course, should be able to do some useful work, but they have limited processing power, sensing capabilities, communication range. Sensing is also could be done only at a limited range, and of course, limited capacity, limited energy. So that means a single nano machine typically would not be useful. So we need a network of nano machines. Then that requires communication between those machines in that nano network. So that there are two approaches in the literature for solving this communication dilemma. One is electromagnetic communications at the terahertz frequencies, and the other is molecular communications. That means communications by making use of molecules. That's what we're focused at. So though we use the title nano-networking, we're specifically talking about the molecular communications implementation of nano-networking. And as I said, there is an IEEE standard group that's working on the standardization of the work in general in nano-networking. But again, the focus of the standard group is also mostly molecular communications, because that appears to be more viable. There are some testbeds that have been developed as proof of concept at the macro scale and also at the micro scale. The one on the top you see was initially based on the sensors produced by Andrew Eckford from York University in Canada, and the overall testbed was published in the given ICC paper. We have a copy of this testbed actually in our lab in Kandilli, but we're also working on some microscale testbeds, as we're going to discuss soon. So what have we done up to now, and what are we currently focused on? First of all, we defined the work I'm going to explain here, by the way, is the theoretical side. That was mostly done by Ali Emre Pusani, Birkan Yılmaz, and myself. Later, Cansu and Sema will be talking on the real implementations based on sensing and others. So, as I said, my explanation will be mostly focused on theoretical work. So, we first defined the energy model for molecular communication via diffusion. This is how we call it in the non-networking world. We designed some different modulation techniques for transmitting data, how you can transmit information by making use of diffusion, by making use of molecules that are diffusing in the environment. We also defined how you can form data frames by making use of molecules. We looked at how calcium signaling is done between cells. And one important thing, I'm going to discuss this in more detail later, but we looked at pre-equalization and ISI intersymbol interference mitigation. That's the major problem in nanonetworking, how we can solve ISI. So we came up with some pre-equalization approaches for that. We also looked at the analytical formulation of the 3D channel characteristics when we have an absorbing receiver, and we also defined the achievable rate analysis for diffusion. We looked at network coding for communication via diffusion and also position-based modulation. And we also defined how a partial receiving, counting, absorbing receiver could be done, and then also make use of optimal reception delay, again, to reduce intersymbol interference. And we also developed some simulation methods and open source libraries like the high-level architecture and GPU programming. We looked at index modulation with Ertuğrul Başar from Koç University. We applied index modulation. Index modulation is already being used in electromagnetic communications, but we were the first to apply it to molecular communications. And we also had a joint project with South Korea, with Professor Che of Yonsei University in South Korea, on communication in vessel-like, like blood vessels, vessel-like environments, how communication and detection also could be done in such environments. So, as I said, all things I very fast went over here are mostly theoretical work that we have done thus far. We currently have an ongoing TÜBİTAK 1001 national project on colorimetric analysis. We have a molecular signal and source localization for underwater and medical applications. That's the project of Birkan Yılmaz. If you're interested in that, Birkan can answer your questions in more detail. So let me very briefly talk about this molecular communication by diffusion, what it is and what are the problems there and how we're trying to overcome those problems. So the problem is Newtonian physics doesn't work, as you know, at the nanoscale. The inertial forces are not that much effective anymore, and we cannot shoot or throw a messenger molecule towards some destination. You just release it to the environment and start praying. Hopefully, with diffusion, those messenger molecules will arrive at the intended destination or the intended receiver. So that means you're making use of Brownian motion. So the molecules you release, hopefully with diffusion, will arrive at the receiver. But the probability that this will arrive in time is very low. Therefore, what you do is you release many of these molecules. These will take different paths. Some will arrive early, some will arrive late, some will never arrive in 3 dimensions, and some will arrive extremely late. This red one here is so late that, as we will see now, it causes some problems. The implication of this delay is that when you release molecules from the source cell, for example, to some destination cell, most of the molecules will go in almost line of sight path and arrive early. That's why at the receiver side, if you look at the number of arriving molecules, it increases steeply in the beginning. But unfortunately, there are also many molecules, there are stray molecules that remain in the environment, and they're taking the long path, as we've seen in the previous animation, and they will be arriving too late. So the right tail of this distribution is a very slow decaying tail. So what happens when you have, now this is for a single bit of information, a 1 or a 0, that's how you send it. If you want to send, and of course you want to send a sequence of bits, the problem is the second, for the second bit, if we plot that with a green curve, there will be too much overlap, this yellow shaded area, which is called the intersymbol interference. These are the molecules that belong to the previous bit, the one that was shown with the red distribution, but they arrived at the time duration of the second bit. That's why it's called interference. The stray molecules, delayed molecules from the previous bit are causing a misunderstanding for the follow-up bits. So this ISI intersymbol interference is the main problem in the nano-networking world, and almost everything you will find will be trying to somehow solve this problem. So the problem gets even worse. Now, that was a very simple scenario. One transmitter, one receiver. What if there are also other transmitters and receivers in the environment that are using the same type of messenger molecules? The problem becomes even more complex in that case. So if we look at the number of receipt molecules at the destination, it will be typically composed of the terms you see here, which are basically the intended molecules, number of molecules that were received at the correct time, and the ones, unfortunately, that were incorrectly received during that time. So these are the things actually that are causing either intersymbol interference or co-channel interference. It's like if you have two pairs communicating with each other, this one is supposed to talk to this one, but unfortunately this one receives it. So that's what we call the co-channel interference. So these are typical problems we need to deal with. So co-channel interference, as I said, has a very detrimental effect in molecular communications. It depends on distance and the distance between the pairs, and also distance between the transmitter and the receiver. So how can we cope with the intersymbol interference? One solution is you put the symbols too far apart so that you decrease the amount of overlap here, which is the intersymbol interference. So if this blue curve was more to the left, there would be more intersymbol interference. Take it all the way to the right, then you will decrease intersymbol interference, but that means you're transmitting even at a lower rate, which is something, of course, the communication engineers do not want to do. One other solution could be you could shape the signal. Rather than having that signal have a very long, slow decaying right tail, make the fast decaying right tail. If you can manage that, if you can do that, then you can have consecutive symbols coming one after another. So that means you increase your data rate. But now the problem is how you shape that signal. So one solution could be you destroy the stray molecules, which is not easy to do, or you try to be more clever. And what we proposed here was, for example, we transmit yet another signal. So actually we want to send A, but we send another signal B, which cannot destroy A molecules, but the receiver makes a subtraction and subtracts the B signal from A. So the resulting signal would be Z, which has a fast decaying right tail as compared to the A distribution. How you implement this is yet another problem. Currently, we're working on that. Professor that's working on synthetic biology. But again, that's completely theoretical and simulated work. One other thing I mentioned earlier was index modulation in molecular communication via diffusion. This is used in the emerging wireless communication systems. Like you have multiple antennas that are transmitting and receiving. Like normally we try to transmit the signal, the electromagnetic signal, and based on some feature of the electromagnetic signal, like the amplitude, phase, frequency, whatever, you modulate the information. This time also the antenna index, the number of the antenna from which you're transmitting, that also contains some information. Like if you have 8 transmitter and 8 receiver antennas, and you understand, let's say you transmitted from the 3rd antenna, that's even 3 bits of information itself. And the information you transmitted, that's another bit. So in total, you have transmitted 4 bits in one single duration. Another approach would be carrying the information at the time of the transmission. So you send a spike, actually, you send a pulse. When you send the pulse, could be the information itself. So if you want to send a 0, you send it at this point. If you want to send a 1, you send it at a different time. When we do transmissions, when we do communications, unfortunately, there is a lot of error in the communication. So typically, we have error correcting codes in communication. So coding is a very important part of communications in classical communications. And typically, we make use of the Hamming codes and look at the Hamming distance. However, Hamming distance does not work when we talk about molecular communications because we're not using the electromagnetic signals here. For this, we have designed the symbol interference minimizing error control codes. Now, remember that ISI figures, we said the overlap was the major problem. So one approach we proposed was, what if we don't start counting at the beginning where we have a lot of interference and start the counting later? But then the question is, when? When do you start counting? How much time you skip that tau time? So we looked at how we can optimize that reception delay and coined the term signal to interference difference rather than signal to interference ratio, which is a better parameter for molecular communications world. The intersymbol interference is mostly due to the molecules that are remaining in the environment, and typically they arrive on the other side because they're taking the longer path. They mostly arrive on the other side of the receiver. So we'll look at what happens if you just start counting on one side, on the intended side of the receiver, and ignore the rest. So we came up with the partially receiving absorbing receivers. And for the 3D channel, the characteristics of the 3D channel were not, actually still not known analytically. So what we did was we came up with the definition of 2 quite complex, one with 5, the other one with 6 parameters, new distributions that represent the channel behavior much better than the existing probability distributions. But setting the parameters of these distributions is quite complex. So for that, we also developed an ANN network. And the famous Shannon's channel capacity formula, unfortunately, that also doesn't work in molecular communications. So we defined and found the achievable rate analysis for molecular communications world. So in total, our theoretical work, Cansu and Sema will continue on the other side of it, but our theoretical work are basically We defined the first energy model for molecular communications via diffusion. We defined several modulation techniques. By the way, we started working on molecular communications in 2010 and 11, which is also when the nano-networking communications society started forming. So we're one of the early bidders. So that's why we also received a lot of hits for these papers, for our early papers. We defined the first frame structure composed of the molecule. We studied calcium signaling. We look at how we can mitigate ISI basically by pre-equalization. We developed the analytical formulation of 3D channel characteristics with observing receivers and achievable rate analysis for molecular communication via diffusion. We developed network coding for MCVD. When you have two pairs communicating in between, you can insert a relay node. I didn't explain that. That can extend the communication range. There we did network coding and also position-based modulation. We defined how a partially receiving, counting, absorbing receiver could be designed. We did work on how to optimize the reception delay to, again, reduce intersymbol interference. And for all these, we have developed several libraries and simulators to this end. So my presentation ends here. So I will pass the screen control now to Johnson.
So please let me know if you can see my screen, then I can continue from here.
We can see your screen, but you have to go to presentation mode.
Oh, yeah, sure. Just a second. I think it's going to be easier like this.
I think we will delay the most of the questions maybe at the end, unless there are burning questions which needs to immediate clarification. Please go ahead, Johnson.
Thank you for the introduction, first of all, Professor Tuju. After this theoretical part, I want to introduce you a little bit what we are doing on the work, like experimental work, because as you know, molecular communication, which actually includes the molecules, it needs to be justified experimentally. Up to now, there was a lot of works on the theoretical part, and there was like really nice things, but when it comes to experiments, as you know, we have a lot of problems. And I think experimentals are going to understand what I'm saying. I will introduce to you different works that we are conducting in micro and nano engineering to support the molecular communication field. To start with the Micron range, as Professor Tuju showed different models, like different simulations, all of the simulations are actually realized inside the body. So it's like a really small size that you are dealing with when you are trying to count the molecules. So the first thing that needs to be done in this field, that's creating an environment, closed environment, where you can study diffusion. And for this, microfluidics is an amazing tool, because this tool provides us to control the flow rate, maybe to reduce the ISI, to play with different parameters when you're doing an experiment. This tool is also nice because by using microfluidics, we can generate different testing environments. For example, when you want to work molecular communication in a certain part of the human body, you can mimic such geometry directly in the microfluidic systems by using like Micron Range models that you want. And for this, there are different techniques, as you know, to produce like microfluidic systems. Laser ablation is one of them. It's quite a rapid technique. It's really cost effective. So you are using only like a laser source. It can be UV laser or carbon dioxide laser. In our case, we are using a carbon dioxide laser to produce a Micron range test environment, as you can see here. This method allows us to produce like two channels without any mask or without any soft lithography, which actually makes us to earn more time, because with the soft lithography, we are only able to produce channels at a really long time. And by this technique, we can produce Micron range channels in 20 minutes, and we can have a closed systems. It's quite rapid. And here you can see different channels that are produced by using different, actually same parameters, but with different methods. We can change the roughness of a channel by just simple pre-treatment process that we are applying. Basically what we have here, we have a PMMA substrate. It's a methacrylate substrate, which we use to create the channels. And by using the laser ablation method, we are able to change the roughness, depth and the width of our channels. So this allows us to produce different test environments. Let me show you one of them. This is a simple device produced by using laser ablation. So we have a Micron Range channel produced by laser ablation. So we are using polystyrene microbeads as information carriers. They are Micron Range systems. So what you see on the right here on the video, we have the polystyrene beads, which are used, the number of the polystyrene beads as the information carriers. So the presence of certain threshold number of polystyrene beads means we have the information 1, and the absence is 0. The red count, let me play again, here is our ROI region, meaning our region of interest. We kept it really short here, so we are using a circle detection algorithm here to count the beads. And by extending your ROI region, you can transfer long sequence bits, such as here is the 14-bit code, which is ASCII letters of Boğaziçi University initials here. We are able to transmit an information effectively by using this method. This is one of our test beds that we are using, for example, when we want to study micro scale information transmission, let's say. Also, we can use microfluidics for off-body sensing systems. What I mean is that when you couple microfluidics by SRR splintered resonating structures, we can sense any molecule that we want by measuring differentially. Here is our system. What we have here, we have on the left a reference sensor, which provides the background data, the reference data, and we have a test sensor. In the reference sensor, we have only one streamline, which we have the DI water in this case, and our model molecule here is a glucose. By injecting glucose with the DI water from the middle channel, we are able to measure the concentration of glucose using vector network analyzer. This method actually allows us to have a real-time detection of the molecule of interest. And of course, non-contact measurement is quite important for us to avoid any contamination or maybe increase the sensitivity. Here is the reflection spectrum of our measurement. Basically, we are able to sense 0.01 molar glucose by this method. But of course, this method is not viable when you want to do in-body sensing, because this is like a really large-scale sensor that you cannot implement directly inside the body. For this, what we can do is instead of doing a differential measurement, we can use nanoparticles as sensing agents. So we have different nanomaterials, like first-generation nanomaterials, we call them, because these materials are not yet biocompatible. This has to be covered with a biocompatibility, it has to be covered with another agent to induce biocompatibility. That's why we call them first generation nanomaterials. And when they are able to be induced, actually implemented inside the human body without inducing any toxicity, these nanomaterials are referred as the second generation nanomaterials. And of course, there is another thing which is important, is the third generation, which we call stimulator responsive nanomaterials, which actually triggers your sensing mechanism. Here we have different geometry, we have different size nanoparticles, and all of them can be used, actually different applications that can be used to sense any specific molecule of interest inside the body. What I mean is that, for example, let me show you an example. Gold nanorods are quite actually most common systems that are used for sensing reagents of sensing for glucose. Basically, in the presence of glucose and glucose oxidase, the results of their enzymatic reaction actually induces a peroxide to the environment, and we can use nanoparticles to sense glucose indirectly by measuring the level of peroxide in the system. Such kind of nanoparticles can be applied directly inside the body by, of course, doing some modulations, as I said before, and they can be used to as sensing agents or messenger molecules inside the body. And this is what we are doing currently for in-body sensing part. And now I can give the floor to Professor Dumanlı for the transmit the information from the body to the outside world. I'll keep it quite short. If you have any questions, just let me know because we don't have too much time.
Thank you so much. We may have a problem with the Zoom ending, by the way. I think the reservation was done according to the actual timing. So if it happens to be so, we should be just using the same link to get back in.
Maybe we can do a little bit quick question and answer time, Can Hocam,
Yeah,
and
maybe.
then reconnect again with the same link. Then it will be on the correct time, maybe, if that works out. I am not so sure.
Probably it will work
We are
out.
trying to resolve this link issue, but it might get interrupted. If we interrupt it, we all reconnect to the same link.
Right. So we have about a minute. If there may be a question to Tuna Hoca or Cansu Hoca. Fast, maybe. Actually, I took a note of calcium signaling. That's one of the implementations of the work, and that sounds very interesting when it comes to muscle work too, because that could be used as a sensor for muscle function. Maybe could you say a few things on that, if I am right, or kind of hallucination?
No, definitely not hallucinating. Unfortunately, that's not that's a field we least focused on because it has been studied a lot in the biology world. So we didn't focus too much on calcium signaling. But you're exactly right. It relates to the way the muscles work. But together with that, actually, you should be considering the neuromuscular junction, and because that's the one actually that triggers the first working of the muscle. One thing that also requires attention there when we start talking about muscles is how the pacemaker muscle cells work in the heart, because that's also something that could allow us to